Okay, good evening, everyone. Thank you for being here in person. It's a full room, which is always good to see. And for the people joining us online, thank you as well. My name is Marike. I'm the Associate Vice President Research. And today we are going to hear about the microscopic world. And to begin, I would like to acknowledge that the University of Manitoba campuses and research spaces are located on original lands of Anishinaabe, Innunuak, and Sinunuak, Dakota Oyete, Dene, and Inuit, and on the national homeland of the Red River Métis. For a personal reflection, being a newcomer to Turtle Island, I'm grateful to be here, and I am committed to building lifelong relationships with Indigenous family members, friends, and colleagues. And I'm also committed to working with Indigenous community in a spirit of reconciliation. So I am grateful to be here, and the Knowledge Exchange is an opportunity for researchers to share their emerging knowledge with the general public and the community at large. The Knowledge Exchange is hosted by the Office of the Vice President Research and International, with support from the Learning for Life Network. And joining me this evening are experts seeking tiny solutions to huge problems, such as viral defense, antibiotic resistance, and environmental contamination. Our moderator this evening is Dr. Marike Palmer, an assistant professor in the Department of Microbiology in the Faculty of Science at the University of Manitoba. And Dr. Palmer's research focuses on discovering new microbes and using genetic data to understand their diversity and evolution. It's now my pleasure to turn things over to you, Marike. Thank you, miigwech, and merci.
Thank you so much, Marike. So as Marike said, I'm Marike Palmer. And just as kind of like an overview of what the evening would entail, of course, each of us would introduce ourselves and a little bit about our background and our research interests. And then we'll open up the floor to questions. And for those of you in the audience, if you want to ask in-person questions, there would be a rotating microphone that you can just put your hands up and we can have those questions addressed. And for anyone watching the live stream, there is a link below the video description that you can actually click and submit your questions to. So please make sure that you make use of that if you're watching online. So I guess as a little bit of more insight into my research interests, as Marike said, basically our bread and butter in my lab is associated with looking for microbial biodiversity and trying to understand what the breadth of microbial biodiversity looks like, how these organisms live and interact with one another, and how we can actually preserve the environments and these communities in which they actually live. So with that, obviously comes the need for effective communication. So a lot of our work actually describes and names novel species or organisms, like new organisms that we might be discovering, in order for us to actually effectively communicate about that diversity. So next up would be our first panelist is Dr. Peter Pelka, and I would ask him to introduce a little bit about his research interests.
Thank you, Marike, and thank you everybody for inviting me to this panel. So I'm a professor of microbiology at U of M here. I've been at U of M since 2012, so quite a while now. My interests are virus-host interactions with adenovirus as my model organism. Adenovirus is a fairly benign virus. It causes upper respiratory tract infection, usually quite mild and self-limiting. It is the major cause of conjunctivitis, viral conjunctivitis. So if you have a viral conjunctivitis, it's probably adenovirus 9 out of 10 times. It causes gastrointestinal infections, etc. So my main interest is how the virus interacts with the host cell, how it changes the cell upon infection, and how it disables the innate immune system. The other aspect of my research is viral-based vaccines. So because adenovirus is a fairly benign virus and easy to manipulate in vitro, it has been extensively used for gene therapy, gene delivery, research, also vaccines. And one of the first COVID vaccines was adenovirus-based. And now we know why there were also bad side effects associated with that particular vaccine from recent studies. So part of my work involves developing vaccines that are based on that virus, but our approach is slightly different. We are interested in vaccines that are orally or nasally administered, that are easier to develop, easier to administer, and reduce hesitancy. In terms of my background, I came to Winnipeg in 2012 to start this job. I was at the time a postdoc at McMaster University. I did my PhD at McMaster. I postdoced with Joe Mimrick at the Western University in London, Ontario, which was a fantastic experience. To this day, we collaborate all the time. And I am very happy to answer any questions about any of these topics and looking forward to the discussion. Thank you.
Perfect. Thanks so much. So next up, we have Dr. Nate Budisa.
Thank you everybody for having me here. I am in Winnipeg about 7 years. Yeah? As you hear from my accent, I'm not Canadian. I'm born in Croatia, former Yugoslavia, and I studied the chemistry, molecular biology, and biophysics. I made my PhD in Germany, and I spent most of my research time in Germany, starting as assistant professor to full professor, and then as a full professor between Munich and Berlin. And from Berlin, I moved 2018 here to be Tier 1 research chair in chemical synthetic biology and xenobiology. My research is unusual genetic engineering, so we are not doing a classical genetic engineering, but we are trying to change rules how genetic engineering works. In other words, we are trying to make synthetic cells. So don't be afraid. This is just normal research. Our laboratory look like all other laboratories. You will find lots of solutions, lots of young people who are mixing solutions and making interesting experiments. So everything what coming out is in our head. So my research has, if I just for general audience, I would say there are 3 layers. First layer is to understand why nature choose this chemistry, these building blocks as we already know it. Because in last 200 years, humanity developed marvelous chemistries. So that means in the terms of chemistry, our human body, our whole life on the earth is just one option. We have many other options. And the question is why nature didn't choose these options. So this is the first question. Second is, by working with or trying to change organism in this way, we are very much concerned with biosafety and biosecurity. And I am one of the father of the concept of genetic firewall. That means the creatures that we are making in laboratory should not survive and could not survive outside of laboratory. And the third one, if we are talking about synthetic life, it's not Frankenstein, it's something, it's actually technology. And this is my research chair is associated with the useful application of this technology in tissue regeneration, or just to develop gene technology one step further to replace, actually to add to this genome editing as we have today proteome or protein editing, which we can talk maybe during this evening in more details. That's enough for now.
Thanks, Nate. Next up, we have Dr. Aleeza Gerstein.
Hi. So like everybody else, I'll say thank you for inviting me to talk here today. I have been a professor at the University of Manitoba since 2018. Prior to that, I was involved with U of M. I was telling my panelists as a child when I attended Mini-U. So I grew up in Winnipeg. I didn't go here for undergrad, but I was delighted to come back here as a professor. During my time away from Winnipeg, I went to the University of Western Ontario in London, Ontario for undergrad, and then migrated over to Vancouver for graduate school. I would say that my training is as an evolutionary biologist, and I kind of fell into microbiology because microbes are incredible to work with in the lab. So we're able to manipulate them in a way that we can't manipulate birds or trees or fish. My general interests came from using microbes to study sort of these very broad evolutionary patterns that we see in nature to try and understand life on the planet, what exists, where it exists, and why. What is it doing there? How is it best adapted to growing there? And how has the evolutionary history of species sort of transpired over time? I've always been really interested in evolution at the level of the genome. So this is DNA, A, C, T, and G, the 4 bases that make up essentially the book that transcribes how we see ourselves or how we turn out the way we turn out. And so my lab uses a mix of microbiology techniques and computational statistics to really understand life at the genetic level. So my focus has always been on fungi. Most people know fungi as mushrooms, which they are, but there's other types of fungi as well, including fungal microbes. And so one of my favorite things about fungal microbes is that we don't know very much about fungal microbes. There's somewhere between 2 to 5 million species on the planet, and we've only described like 300,000 of them. The ones that my lab tends to study recently are the human fungal pathogens. Candida albicans is the most common. This is the bug that commonly causes vaginal yeast infections. If you have a vagina, you have a 75% chance of having one of these infections during your life. And so this has been a focus of my lab. This is evolution in action in the human body. And over the years, I've gotten very interested in the microbes that don't just kill you, but also the microbes that cause significant morbidity. So these things that cause these chronic infections, and like much of women's health research, we don't really understand them very well. And so we're trying to understand diversity within the human body, what different fungal populations look like or how they differ between your mouth, your stomach, your skin, because it's the same species that live at all these different body sites. They often are growing there. Many of us in this room have these microbes in us, and they're existing there happily. You don't even know they're there. They might even be doing beneficial things, but then something can flip and they become pathogenic and they start causing these symptoms. And so that's sort of broadly what we look at, amongst other things in my lab, including things like antifungal resistance that lots of people like to study as well.
Perfect. And then our last panelist, but definitely not least, is Dr. Ayush Kumar. So over to you, Ayush.
Yeah. So it's a pleasure to be here. Wonderful to be sitting with my colleagues here and talking with you all. So as Marike said, my name is Ayush Kumar. I actually did my PhD here, so I'm a product of U of M, and then I was gone for a few years. So I did my postdoc in Colorado, working on some of the bacteria. So I worked with really nasty bacterial pathogens. So for my postdoc, I actually worked with organisms that are potential bioweapons. And then I started my own laboratory in Southern Ontario. And then, after a few years, I'm like, well, what kind of place is this? You don't even have to plug in your car. You need to get back to Winnipeg. So I just came back to Winnipeg. And, so I've been here since for past 15 years or so now. So as I said, we work with some of the nastiest pathogens, bacterial pathogens that are out there. So WHO, which is the World Health Organization, every couple of years releases a list of bad bacteria against which we need therapeutic interventions urgently. So 2 of those organisms are the organisms that we work with. One is called Acinetobacter and the other is called Pseudomonas. And in particular, what we are trying to understand is how these organisms become resistant to antibiotics. So antibiotics have been a miracle of modern medicine. So since their discovery, which was less than 100 years ago, it is estimated that antibiotics, at least in developed world, have added about 20 years to our lifespan. So we all live about 20 years more than we would have in pre-antibiotic era. But this is changing rapidly, because bacterial pathogens are becoming resistant to antibiotics. So we study, and there are various mechanisms by which bacteria become resistant to antibiotics. So we study a kind of protein that bacteria produce, and what this protein does is it captures the antibiotic molecule before it can get to its target within the bacterial cell and then pump it out. So as a result, bacteria can survive in presence of antibiotic. So what we are trying to understand at molecular level is, first of all, how does that bacterial cell know when to produce these proteins? So what are the chemical signals? What are the environmental cues that it takes into account to produce these? And then we are also trying to understand how these proteins function. And the ultimate goal is to use this knowledge for therapeutic intervention. So probably find some new antibiotics that can bypass the activity of these proteins. Some of the other work going in our lab is also looking at the prevalence of antibiotic resistance in environment, and this is one work that we have actually done in collaboration with Dr. Annemiek Verrenhorst. For past 15 years or so, we have been working with several First Nation communities in Manitoba, studying their drinking water and source water, and analyzing their water for presence of antibiotic-resistant bacteria. And I think when I first started working on that project, first few results that we got, I was very, very surprised to see how poor the quality of drinking water from First Nation communities is, and how, as Canadians, we can even accept that. So, and that has been story over and over and over again, unfortunately. And then we also work with some industrial partners trying to find novel ways to inhibit bacterial pathogens, particularly in hospital settings. So, yeah, I'm really happy to be here. I'm happy to answer any questions you may have. Thank you.
Perfect. Thank you so much. So anyone that has questions, you're more than welcome to just put up your hand and we can address those questions as they come in. But maybe as a starting off point, we've obviously got a mixture here of bacterial, fungal, viral, synthetic, all the way through in terms of the biological potential for threats. So what can we do potentially to effectively assess the potential threats posed to humanity and to environmental health in the context of what these different microorganisms look like? Does anyone want to go first? Because I'm sure we all have opinions.
Maybe I can start. So I think my 26 years in Germany and now 7 years in Canada show at least both countries having same problem. There are three levels of the problem. First, science itself. So do we understand what we are doing? Do we think about consequences of we understand, of course, what we are doing, but do we think about the consequences of what we are doing? This is the first question. Second question is outreach. How our scientific achievements in laboratory are presented in broader audience. I think this is not difficult. I mean, good science could be you could explain I think in understandable terms. It's the purpose of science is actually to provide understanding on the level of technical language, but also the level of language of general audience. And the third is of course citizenry, your citizens. So especially this what I call enlightened citizenship. So, and I think when we look historically, for example, in Germany, look at the German Green Party in their level when it was emancipatory power. Today they are establishment. So what they were, for example, in 1980s, they were furiously demonstrating against recombinant production of insulin in Germany, because it was a gene technology and it was something evil. But now, 40 years after, many of these people who were demonstrating against recombinant insulin, taking this recombinant insulin, exactly, they are not taking animal insulin because it's more efficient, it's more clean, and it's actually a more human way to produce insulin. And what you see the problem here, the problem was communication. And I think this problem is still unresolved. And we need such more meetings of such time that we actually communicate, that we understand whether we are, whether we have a language of understanding.
So I would add on to that. I think understanding risk and understanding uncertainty and what isn't most likely to harm you from a microbial perspective. So I have 2 young kids. We eat food off the floor all the time. Like, we live by the 5-second rule. When we're camping, they eat things off the forest floor. We don't worry about food that's dropped. I'm personally far more concerned about the things that we don't see in the air. I still regularly wear a KN95 mask in public spaces. And so for me, it's understanding and also advocating for funding where we can actually make a difference. So most of us in Canada, again, accepting some certain communities, which we should deal with, don't worry about our tap water, right? You've probably never, ever been concerned turning on a tap in this country and drinking a glass of water from the tap. That's because in England, a long time ago, polio was transmitted through the tap water, and we advocated for clean drinking water. We no longer worry about the clean drinking water. I was hopeful that we would be advocating for clean air in a similar way, and it has happened in some places. We have new buildings in the science complex at U of M, which have phenomenal air quality. The air filtration system is good. It mixes over fresh air constantly. But we haven't had this like mass push to clean the air. And anyone who has young kids know that they get sick all of the time. Some of this is because of not washing their hands, but some of it is just because the air that they're breathing is not clean. And so I think that is an education piece. And it's also, sometimes the science is really far away from where the policy lines up.
As a virologist, I think the biggest problem is part of it is obviously, as Ned mentioned, and my other colleagues as well, is communication, where lack of communication and misunderstanding of what is in a treatment, what constitutes a risk, is a major problem. And I think that's perfectly illustrated by the resistance to the mRNA vaccines. Everybody who was resistant to it did not understand they don't even know what mRNA is. They don't realize every time they eat anything in their food, they're eating tons and tons of mRNA, as well as DNA and everything else that you find that is quote unquote artificial. And communicating that, I think, is very critical. But also, I think that lack of understanding and this sort of Twitter or TikTok-based education campaigns where somebody's friend said something and that's a fact because, scientists are evil and they're the establishment, cannot be trusted, right? And that leads to ignorance, and that leads to, what we're seeing now is outbreaks of measles, a disease 10, 20, 30 years ago was unknown to the developed world. We never had measles outbreaks. When I was growing up, when most of you were growing up, or at least were adults and had your own children, I don't think you ever worried about going anywhere and worrying about measles exposures. But to look at a different perspective as well in terms of identifying threats that can be potential threats from a pandemic perspective, like COVID, how did that come about? And how do you identify those things early? I think it's a difficult problem, and we don't know what is bad until we see it, right? But I think trust in science, continued funding of science, funding for surveillance, international collaborations is important. To this day, we really don't have a clear picture of how, for example, COVID emerged because of secrecy in communist China, right? They are, centralized government, and they don't want that embarrassment, they are getting blamed for it. That affected the entire world in a dramatic way. Our lives would be completely different today had COVID not happened, right? So, it's communication, it's education, it's collaboration, it's recognition of what can harm us. And to do that, we need all those other things in place. Without them, we are living in ignorance, And then we have outbreaks of polio. You know, it's a big problem in New York because of fairly conservative Jewish populations that do not believe in vaccinations. We have measles outbreak all over. You know, Canada has lost their measles-free status last year. US is going to be next. And this year in Manitoba, so far, we have more cases than we had in all of 2025. These things grow exponentially. So this is just going to become worse and worse. UK, meningitis outbreaks, all preventable diseases. And that because of ignorance, lack of understanding, not trusting in scientists because they're establishment, etc. And we have to collectively fight that to overcome that problem. And it's going to be, I think, a growing problem for us.
Yes, just to add to that, what keeps me up at night is in our lab, we have bacterial isolates that came from patients in Winnipeg, from other parts of Canada, that do not respond to a single antibiotic that we have in our arsenal. Not one antibiotic. And I always wonder, if somebody gets that infection, what is going to happen other than watching them die, right? And we have seen that. Recently, I was talking with a colleague who was talking about a case, a 7-day-old baby infected with an organism called Klebsiella, not responding to single antibiotic. And there's nothing you can do about that, right? And so antibiotic resistance, it's a silent pandemic. It kills around 5 million people around the world every year. But, we often don't hear about it. But as my colleagues were talking about communication with public, because I think we in our labs can only do so much. I think what is important is for everybody to start, and when we talk about trust in science and trust in scientists, that doesn't mean you just listen to things, but ask right questions to right people, right? Don't go to just the Twitter and just see the first thing that shows up, or, when you go home today, try an exercise. Type in word vaccine on Google and see what shows up right as top hits. And those are all anti-vaxxer sites. Don't trust that. Just look at what is actually reality. Ask questions. We are here in Manitoba. When it comes to microbial research, we have some of the top people at this university. Ask them, right? We have team in university with communication team. Just ask the question and try to get right answer and see if you can get convinced by that answer. Because otherwise, I think one of the problems that we have, science is slow. So, COVID vaccine was a miracle. That really doesn't happen. Things don't really happen that fast. And then we are always chasing the negativity around success of science, right? So antibiotics is a perfect example. They were discovered, they were used widely, and what did we start doing? We started using antibiotics on our chicken. We started using antibiotics on our orange orchards. We started using antibiotics on cattle, right? Why on chicken? Because it helps your animal gain weight faster. So in 1940s, to get a chicken ready from farm to market, it took 89 days. Can you guess how many days it takes today? 36. 36 days, you get enough meat in that chicken because of use of antibiotics as growth promoter to put it out in the market. So when you're actually, buying chicken, buying meat, if you're not being careful, you may end up buying meat that is contaminated with antibiotic-resistant bacteria, right? So sometimes what happens is that these scientific discoveries, which are wonderful, but if we don't know how to use them wisely, then we are going to mess everything up, right? And there's no way to fix that. So yeah, I think that's the problem that keeps me up at night.
Yeah, similar story from the fungal space quickly. There's a species called Aspergillus that you probably haven't heard of. Again, common, we're all breathing in Aspergillus spores all of the time. And most people in this room, unless you're immunosuppressed, no problem. In Europe, they're finding people coming into the clinic, so human clinics, with multi-drug resistant Aspergillus infections. So they've breathed in the spores, and they're coming in with an infection that is already multi-drug resistant. They've never taken antifungals in their life. This is because of the tulip trade. So they spray so much antifungals on tulips to ward off the fungal pathogens that they're selecting for resistance in the environment, and the drugs we use in the field cross over with the drugs we use in the clinic. We have very few drug classes for antifungal drugs. If you've had an antifungal skin infection, you know they're really hard to treat. And so it's like this misuse of drugs is a very big problem.
Yes, hello. This question comes from Donna, who's watching online. It's directed to the full panel. Do you use microscopes in your research? And what other morphological, chemical, and other analysis tools do you use?
Who wants to start off?
So I am chemist, and I'm using microscope.
Yeah, I think we all
So
love microscopes.
why we are using microscope? Because we are trying to get our bacteria to grow on some stuff that does not come in nature, and then we look how bacteria react also in the terms of morphology, and then normally we found them they are a little bit slim, elongated. So I mean this is just microscope is something what makes biosciences. Yeah. It's a universal tool, and we will always use it.
We also use microscopes. Fungal cells are bigger than bacterial cells, so it's maybe easier. We have what's called an EVOS in my lab, so we can take a 96 well plate, and in each well, we can scan each well individually, and so we can do things in really high replication. And we use this microscope for several reasons to look at the cells for morphology. Fungal cells grow as these single cells, so that's a yeast cell. They also filament, and they can also form biofilms. And so we use that microscope to do biofilm assay experiments, where we see how quickly they form biofilms or whether certain drugs prevent them from forming biofilms. But the coolest thing I think we use it for is what we call as competition experiments. So we can put a fluorescing marker, often it's called GFP, into a cell. And so the cells that carry that marker glow green. And we can compete the green fluorescing cells against any other cell we want, and we can track the ratio of green fluorescing cells against the non-fluorescing cells over time to measure their ability to compete, essentially. And we can do this in 96 cell plates.
As a virologist, you may think that I wouldn't use a microscope because viruses are not visible under regular light microscopes. You need high-powered electron microscopes to see them. But we use microscopes extensively. We have 3 microscopes in the lab, plus we use additional microscopes that are available at the university for studies of how the virus changes the cell. You can see changes to the cell upon infection within a few hours. We call that the cytopathic effect, or CPE for short. And you can see how the virus spreads by looking at presence of viral proteins, how the cell is changed by the virus morphologically, and how the virus kills the cells. We do additional assays using many other pieces of equipment and studies. We quantify viral DNA and RNA as the virus replicates in cells. We look at how viruses affect cellular gene expression using things like quantitative real-time PCR, which allows you to quantify nucleic acids, whether they are RNA for gene expression or DNA for virus genome replication. We use various protein-based assays looking at presence of viral or changes to cellular proteins during infection, and many other assays. You'd be surprised how many different assays you can do in a lab that studies just viruses, which you can't even see with your naked eye.
Yeah, we use microscope as well, maybe not on daily basis. We primarily work with DNA and RNA, and a lot of genetic manipulation in these bacteria that are resistant to antibiotics. So for example, we have created some bacterial strains where we have deleted all genes that encode for these proteins I was earlier talking about. So these cells are highly susceptible to antibiotic. And the reason we have this cell in the lab is so that we can test potentially new antibiotics against these cells, just to see what are the molecules that will be affected by the activity of these proteins and what would be independent of that. But we do use microscope to study how changes in the antibiotic resistance in the bacterial cell affects their morphology. So sometimes what happens is when bacteria become resistant to antibiotic, they're not very happy. So we are actually forcing them to become resistant to antibiotic. When you take the antibiotic away, they sort of go back to their normal lifestyle. So these are some of the changes that we can observe under the microscope to see if resistance can cause morphological changes in bacteria.
And I just wanted to add to, in terms of the analytical tools, we've actually got numerous labs having capacity for genome sequencing, RNA sequencing. So like actually looking at the nucleic acids of these organisms themselves, determining the sequences of that, and then actually looking at what changes happened in their DNA or in their protein expression process, what might have changed in those particular cell lines. So I just wanted to add that to the toolkit.
Maybe one more thing we have in my lab is called a flow cytometer, which lets us measure genome size in our fungal population. So humans all have 2 copies of their DNA. You get 1 copy from your mom, 1 copy from your dad, and typically you have exactly 2 copies of each of your chromosomes. So most deviations from that exact ratio are inviable. Fungal cells are very tolerable of extra copies of their chromosomes and differences in their base number. So we have cells in my lab that have 3 copies of their chromosomes, 4 copies of their chromosomes, 4 copies minus 2 plus 1. They do all kinds of wacky things. And so we use a dye that essentially like goes into the DNA, and then we use this machine to measure essentially how much of that dye is in each of the cells, so we can actually measure the genome size of our cells.
Perfect, thanks. My name is Tanner Devlin. I'm a process engineer. I do water and wastewater treatment. Great discussion so far. I kind of have a question. It's based off of where we were before. So Dr. Kumar, you were talking about it a little bit. So I think there's no question that there is a line between clean and not clean enough. I think a question for each of you, is there a line between clean and too clean, and where is it?
Right, that's a very, very good question, and it's an important question that plays a role in certain things and pathologies in people, particularly allergy development and immune training that can lead to inappropriate immune responses. And sometimes you hear, oh, this 27-year-old athlete got influenza and dropped dead because of cytokine storm, right? And why does that happen? And there's many hypotheses about that, but generally your immune system needs to be trained to properly respond to a pathogen and be able to recognize a pathogen from a harmless, pollen grain or something like that. And trust me, I suffer from allergies myself, and I know I didn't grow up in a clean environment. So I think, as long as you don't get sick from eating dirty stuff, you're okay. Too clean is not good because you don't expose your body to that, early, antigens or particles that can cause disease or can train your immune system. And then the immune system doesn't get trained properly, and then generally what it does is it overreacts because it has not been shown how to properly train. That's why when you have a child, they go to daycare, they get sick, they get 40-degree fever, and you panic, as a parent, because that's, it's new to them, and the immune system is learning how to properly respond. Now, as you get older, your fevers don't go so bad. And that childcare experience, for example, that eating off the floor, that playing in the dirt is very important in establishing proper immune responses to infections and training the immune system not to overreact when you get a mild infection or a more serious influenza infection. So like I said, as long as it doesn't make you sick, it's okay. I wouldn't go out eating off the barn floor perhaps, but in your house, if you drop it on the floor, you pick it up, I think it's totally fine. And as long as you don't get sick, you'll be all right.
Yeah, that's a wonderful question, actually. And as Peter said, too clean is not good for health as well, right? So, and there is enough data now to support that. So one of the most common, and Aleeza might be able to speak more to that, is allergies in people born through C-section versus vaginal birth. So vaginal birth exposes the newborn to good bacteria, and C-section does not. And rates of allergy are about 20% higher in babies in their adulthood born through C-section. There's also enough data that if you have pets at home, then you tend to pets. So you tend to have lower case instances of allergy. So, and this is something that we are just starting to understand, right? And again, just going back to my interest of antibiotics, in the past, you find bacteria in something, you treat it with antibiotic, you get rid of that. And now we are realizing, no, there are good bacteria and there are bad bacteria. We really need to differentiate between these. Because if we don't, then we do. Good bacteria that we carry are actually part of our immune system because they keep training, as Peter said, our immune system on a regular basis. And when you take that training concept away, then then our body the immune system really doesn't know how to respond to some of these infections.
It's okay. Yeah, it's okay. You mentioned about chicken. Sorry. It's okay. Yeah. About chicken. What about chicken eggs?
Okay, so
About chicken fed by antibiotic. What about eggs?
Yeah, so the question is, for chicken that are fed antibiotics, what about eggs? Eggs are known to contain antibiotic as well. Yeah.
Maybe I can generalize answer on your question. So the question was, what is clean, what is not too clean? So I answer with proverb of my Croatian folk, who simply say, one hair on the head is too less and one hair in soup is too much. So what does it mean? In biology, everything is context. So you are making microbiological experiment. You need to have a clean bench. No sports, nothing. But when you are growing up, and if your parents trying to keep you under the glass bell, then of course you will be later having all possible diseases. So biology is context.
Well, thank you for the start of this great conversation. I think it's a very important question what you raised at the start, because this huge gap between the community and the science field, it's just growing. Moreover, you are losing this field to the influencers and those who fake the results. They earn money, but money are invested here. And my question is following. Don't you find that the reason for that is the conservatism of the scientific community as well? Let's say I have a product and I'm developing it in public health to prevent community from deteriorating from chronic diseases. If I write you a letter to collaborate, it will take very long time because you're all busy with your studies, researches, and everything. And that's fine for people. You're boiling in your field, receiving money to invest your research, to raise these questions. It's quite away from the community itself. So the community is not influencing that way. So there is no any breach actually, neither between the community nor entrepreneurs nor University of Manitoba or any other scientific field. Thank you.
I can say, at least in my world, so writing grants to CIHR, so the health agency to study recurrent vaginal yeast infections, I have a participatory action research plan that involves people from the community feeding back into my research. So part one of my grant is form a committee of 6 people who are not academics, to participate in every aspect of the research program, to hear from them, from their lived experiences, what they want and what they need and what their insights are. Because people with this health condition know better than me, I think, in many respects. And there's a large push, at least on the health research side, to involve people with lived experiences and who have ideas about what to study in the research. That maybe is different from what you're saying, but that is something that I've seen that's, I think, being heavily promoted right now in the health space.
Maybe I can say, so trust the science. I don't like this term, you know. First of all, about scientists. I think in my life, I never met such conservative people as scientists. These are the most conservative people that you may imagine. Even the Vatican Curia, Inquisitia are a liberal institution in comparison with academia, if I may say like this. But think about, we are very much limited with the funding that we have. We are very much limited with also with our time. I have only one head, 2 hands, 2 legs, and I receive daily about 100 emails. After my spam, after filter spam, and I'm, you know, every day I'm running, every evening, falling asleep, trying to answer emails. And what happens? Happens when if I do not reach your email immediately or so, then it takes 1, 2, 3 weeks, 1 month, and then I answer them. I said, oh gosh, this is some beautiful Sunday after the sport, and then just look and I say, okay, these are the emails. And then my email, normally always I apologize for my delay answer. This is my most used word in my emails. This is the first thing. Second thing about losing against influencer. I said, I don't like when people say trust science. Why? Because the science is not absolute truth. And what in science we have, and you can see in some disciplines, for example, origin of life, there are lots of gaps in our knowledge. And then you have populist influencer which will exactly use this gap and say, oh, you cannot do it, you cannot do it, you cannot do it. And that's the problem. And then influencer, they are using language which is more appealing than our language. That's the reason my criticism comes, is also that that's always problem, how to communicate science, that we keep truth and originality of our message, but at the same time that we are not making show. If you make show, then you lose your credibility. But unfortunately, often in our grants, we are forced to promise blue on sky. We will remediate environment. We will eradicate cancer. We will do, and then you should write this. If you don't write this, your grant will not pass through. So there are lots of very, very unfortunate things. And we are kind of fringe minority in society. So if we are now making here a strike because our funding is not increased in last 30 years or something like this, nobody cares about this, because it's here some people coming looking jets and they see there is some University of Manitoba. But if Winnipeg bus drivers make strike, then we will have influence. Yeah. So that's the now question. And the question is, of course, how we can, in the communication gap, I already talked about this, and I think one can work on it, but the question how we can keep society like Canada, like West Europe, like free world, how we can keep us resilient. The solution is very simple. We now say increase funding, but at least keep funding with the level of increase of life costs. And then when society invests a little bit money in the knowledge, if you have in Manitoba, you say, why do I need Peter Pelka and his viruses? You don't need this guy. You can just make optimization and throw away him, and then it comes one pandemic, and all knowledge about virology is gone. And then you keep knowledge about genetics of fungi. We remove Aleeza, say it's not economical to have her here, and then we have some fungi, some spores, some fungi pandemic fall on Canada, and you have no knowledge. And that's actually the university is a kind of knowledge keeper of society. And if you don't invest in your knowledge, and then of course the knowledge will be gone, and then you cannot be prepared to react, and you cannot predict. Not serious person can tell you what will happen, which pandemic, which catastrophe is going to us. Of course, it will come some catastrophe. Human life is problem solving and confronting with catastrophe. Therefore, keep post-secondary education is also question how political elite is capable to think strategically.
So to answer your question, I think you're right. There are a lot of barriers when it comes to for us to work with partners coming from outside university. Things are changing. They're slow. I mean, communication was brought up. When I was a grad student, 26 years ago, I was never taught about science communication. That wasn't even on the radar. And now we are, our students are learning that. But there are, in terms of partnerships, there are challenges as well, as everybody said, because we have commitments often in the lab. We have students who are pursuing their degrees. We have postdocs who are pursuing their careers. So the pivot is not that easy. That, if somebody says, hey, you know what, I've got this wonderful solution, which can be an antibacterial. Can you have 2 people in your lab working on this? It can be tricky at times if it's not related to their project or if it's not related to the grants that are funding. However, I think University of Manitoba is actually starting to do very many good things in terms of fostering partnerships between industry. And so one of the things I think for community members to know is who to talk to if they want to have a partnership. I think sometimes cold emails to a professor are probably not going to be as effective, but if you go to the right office, then they can certainly introduce you to a group of people who might be able to help. Now, funding is definitely an issue, and I think this is where, one of the reasons why I agreed to sit on this panel is that I think we as Canadians have to sort of think about how important science and post-secondary education is for our society. Right now, among G20 nations, if you look at GDP, GDP, the proportion of GDP that goes to research and development in Canada, we are dead last. Number 19 is Italy. We are dead last. So, if we are okay with that as citizens of this country, then fine, let it be. But if that's a problem, because how do we prepare ourselves to, for next time there's a pandemic, can we even produce vaccines? Because we were completely dependent on everybody else. And I think what we are seeing right now is nobody is nobody's friend. You have to be self-reliant. So I think the question is for all of us, right? Do we want investment in this, in science and technology, science and research and development, or not? And then the avenues will open up.
Can I quickly jump in? Sorry, I also just wanted to say, because I don't know that everyone, in terms of general public, necessarily know this, but the vast majority of our actual findings in the context of scientific findings is undertaken by graduate students. And the graduate students are dependent on stipends, and all of our graduate students are underpaid. Like, that is just a fact, because we never have enough funding to pay them.
Please don't take it as a critique.
No, of course not. No, of course not.
Right.
It's absolutely a necessary conversation, but I think that is also one of the really big problems.
Right, and my colleagues, I entirely agree with them, and they make excellent points. Now, sort of to add to that, there has been some change and push to increase industry, academic collaborations, and integration and engagement. For example, the CBRF grant that I'm a director of, that's a critical component of that funding, and we have to really engage with industrial partners, government institutions, and others to build those partnerships. But like my colleague says, generally it is difficult because of contractual obligations, funding obligations, timelines, IPs. But we've certainly done quite a bit, especially during COVID and since COVID, where we have worked with private firms to do some research for them on a fee-for-service basis, or they funded a graduate student or some kind of project. But the other thing I think that is important to mention is that basic discovery research is really what drives innovation and what has led us to the world we live in today. And funding organizations like NSERC that are not driven by health improvement, developing some widget that does A, B, or C, or some gadget that does something. That's not how discovery has led us to this current world. It's basic research. I'm curious about why this happens, and I want to know why. And that's what has really driven innovation and led us to many of the greatest discoveries that we had in the last 100 years of modern science. So that is still very important, and we shouldn't lose sight of that, but it should be along collaborations with private organizations, private companies, government institutions, and others.
And can I just add one more thing to this? Because I want to talk about influencers for 1 second, because I have a few colleagues and friends who are scientific influencers, and they're very successful at this. One of them is employed by the Perimeter Institute in Waterloo, so she's an astrophysicist. 50% of her job description is essentially being an influencer. So that is baked into her job, and it is baked into her evaluation that she does science. But 50% of the time she's not teaching, she's not doing service to the Institute. Her service is essentially being a science influencer. It is not part of our job description, for better or for worse. And there have been several times in the last pandemic in which I have spent considerable effort doing sort of outreach on social media. This is at the expense of my actual job or at the expense of my family. And so that is also a value structure problem.
Yeah, thanks. I appreciate the discussion. I had a quick question that kind of ties into some of what we're talking about here, and I guess that is, you know, the socio-political climate these days and the US withdrawing a lot of funding for basic health research or basic science research. I've always been curious, does that sort of affect universities in Canada a lot? And then I guess what does the future look like from you guys' perspective?
That's a good question and very good point. And certainly it does impact us here as well. It impacts the entire world, not just us, because US has historically been the driver and the biggest innovator. They also take in a lot of our grad students. They definitely love Canadian PhDs because they're very well trained. And a lot of Canadians go there, get good training as postdoctoral fellows, and come back here. And the damage that is being done is absolutely epic, and it will take generations probably to repair it. The level of funding at NIH has dropped to levels it has never been so low before. The ignorance of the US government and the Secretary Kennedy is beyond the pale, to be honest with you. And the word salad that come out of his mouth thinking he is speaking as an educated person is gibberish if you really listen to it. But, you know, in terms of direct impact on Canada, beyond, you know, reducing opportunities for our grad students for training, as well as training high-quality postdocs there that can come back to Canada and do research, we get initiatives from our government and that aim to attract, you know, scientists from the US and give them funding. But that's a very short-sighted view as well, because we are, as was mentioned earlier, extremely underfunded here. For example, CIHR success rate has dropped from over 30% in the early 2000s to below 15 currently. So you have more people applying for less money, and these grants have not increased in funding for probably about 40 years, to be honest with you. For example, NSERC in the 1980s, average NSERC was $30,000 to $40,000. Today, the average NSERC is $30,000 to $40,000, except it was in 1980s dollars, so it went a lot further. You could have a postdoc, 2 techs, grad student, that you could fund with that. Now you can barely fund a single grad student. So spending on these Canada Plus or whatever they're called researchers, $1.5 billion, I think, that could pretty much double the entire Tri-Council funding, including CHR, NSERC, and SHIRC. But dedicating it to only new recruits who will come in, compete for the same funding we're already competing, you know, and further reducing investment and funding success will definitely have dramatic effect. I'm not even talking about the scientific carnage that is happening in the US that will have truly global implications.
So both our federal and provincial governments love to fund what I call press release science. So this is where they say things like, we're going to invest $5 million in AI or whatever the buzz thing is of the day, rather than just investing the money into our very functional Tri-Council, which is SHIRC, NSERC, and CAHR. The Tri-Council in Canada is fantastic. The way it's evaluated is, by and large, I think, agreed to be very equitable and fair, except that the budget literally needs to be doubled as a starting point. So I crunched the numbers on NSERC. The median grant is indeed $40,000. This is the NSERC rate for a single PhD student. So that is, you can fund one PhD student off of the median grant, which means fully half are below that. And that's zero money for research, zero money for sending that student to a conference, zero money for upkeep of your equipment that that student is supposed to work on. And that has not changed. The other, and for publications. The other thing I would say is I know of at least 2 people in Winnipeg who had NIH funding, who overnight had their grants taken away. And I have relied on American colleagues to fund my cooler science. There is no way, there has just been no pathway to fund cutting edge science in Canada without having American colleagues for a very long time. And even though the NIH has been annihilated, they are still outfunding us by a mile.
So maybe I can add my experience, since I spent most of my time in Europe. And when I compare Canadian system, university system, and German university system, you will be surprised. The Canadian system is better in terms of organization. If you are better, you guys are better organized than Germans, and Germans are kind of better for organization. But the problem that you have, if you would invest 1/3, I was German investing in data science, you would beat Germans. Why you are not doing this? I think first step in this direction would be, for example, this ERC, European Research Council. Canada should stick more on Europe, invest in this Europe, because these are very, very highly competitive institutions, and the bank can get, I think, good funding. I think we already started here to apply these grants. So not look only on cowboys in South, but you can look also over the pond. There are people which are culturally and in any other terms very similar to Canadians. Nice people.
We have a lot of people waiting to ask more questions. So here's the next one.
Thank you, guys. It's been a wonderful discussion so far. My question was on something that was that you guys mentioned earlier. So air quality, and it was mentioned that the fertilizers that were used on tulips had started to affect the air quality. And so my question was, based on the research that you guys do in labs, do you think something to that extent and misuse of that could have been predicted earlier on? Or is it just a lot of the times we end up trying something and then it just either goes right or either goes wrong? But I don't know if you guys can answer that, but.
I could say I think we've known for a long time that we're misusing antimicrobials and antifungals on the planet in agricultural spaces. And yeah, I think we know that. I think that's well known. And I think we can't always predict what will happen in the future, but I think we know that if we dump a bunch of antibiotics into the environment, we're going to end up with antibiotic-resistant bugs, and they're eventually going to end up in humans.
In terms of air quality, we've known that air quality is important to health for a very long time. For example, during the Spanish flu outbreak in 1918, it was well known that air quality or stale air was responsible for more infections. That's why they had open, outside classrooms when possible. Even in wintertime, they would have classrooms that were under a gazebo or something like that, because they recognized that. But because it's a costly investment to improve air quality, And many buildings are old. For example, my building where we work in, most of us, except Ned over here, is Buller. It was built in the 30s. You know, it has constant air quality problems, but it's very expensive to resolve that. Ideally, you would have multiple, 8 to 12 air exchanges per hour in a building. But to achieve that, to reduce infection rates, spread of, allergens and other things, it's expensive to retrofit old buildings. And you get by with what you can, right? Certainly. But it's like I said, it's been known for probably over 100 years anyway. It's not a new problem.
Hi. Oh, sorry. I've never done this before. Okay. So this is for...
Us either. We haven't done this before either.
Oh,
so.
we're in the same boat. Okay, so this is for the virologist in the group. I think that was... Yeah. Sorry, I forgot your name. What's your name?
Peter.
Peter. Hi, Peter. Okay, so this is going like way back to the beginning of the presentation. In what ways can the innate immune system be disabled by adenoviruses, I think you were saying? And I have follow-up questions. So I'm looking for things like MHC class 1, surface presence on cells, regulatory cytokine reduction, and like interferon reduction. And also, how do these specific mechanisms impact the immunogenicity of those cells?
Right,
Yeah.
well, that's right up my alley, so thank you for the question.
You're welcome.
The simple answer is adenovirus can do it all, as do many other viruses. So it blocks presentation of antigens by MHC class 1 molecules on the cell surface. It causes internalization and loss of those molecules. It directly inhibits production of interferon. It inhibits ability of cells to respond to interferon and produce the interferon-stimulated genes. So these are proteins that will be made from those genes that block virus replication. Essentially, it can do it all. Many viruses do it to some degree. In terms of, how does it influence antigenicity? It does reduce it, at least initially, and blocks, prevents early detection of the virus. So at early stages of infection, the virus hides away, and you are always most infectious before any symptoms set in. So you are usually infecting others before you feel sick. And when you feel sick, when you get that malaise, the headache, the fever, that's actually your innate immune response. So production of cytokines, interferon is what makes us feel sick. At that point, the system recognizes, your immune system recognizes you've been sick, and responds to it, virus titers generally start to drop very rapidly because interferon is generally quite, effective at blocking virus replication. But the virus does it early enough to replicate enough to cause enough virus to be produced, to release. But once that infection has spread enough, there is enough antigens produced and other things that it does not affect sort of long-term protection, memory B cell production. But that takes, 2 to 3 weeks after you've been infected. So it's that innate immunity that protects us. Initially, it reduces the virus is able to reduce initial innate immune response, so it can replicate enough to infect another host. But in terms of long-term protection and humoral protection, so your protective antibodies, it usually doesn't have that much of an effect unless you get measles, which wipes out your memory B cells, and you go back to as if you were never immunized. So that's why measles is pretty nasty, and we should all try to get immunized. Sorry? HIV generally infects T cells. Yeah. So it disables sort of the effective arm that sort of executes the immune response and does a lot of the hard work, like cytotoxic T cells, and other things that drives the immune system response. Yeah.
We have another question from Donna online. This is a question for Aleeza. Do you study genomic variants in Candidus albicans and other fungi, hopefully you know what that means, and other fungi that interact with humans? Is there variance, or are the sequences stable?
Yeah, similarly technical question. So yes, that's exactly what I do. So when you go to a clinic, let's say you have a fungal infection. So the clinician, pathologist will take like a swab of whatever body site is involved, and they'll send it to a microbiology lab. And at that microbiology lab, they'll take the swab and they'll plate it onto like special media that the fungal cells like to grow on. And typically what they're doing is looking for, something to grow, and then they'll figure out what species it is, either because it grows on one specific type of media versus another, or we use this cool thing called chrome agar in the lab, where different colors mean different species. And once they get to that, that's typically it. They take one colony, they're like, this is the species, end of story. But in my lab, what we do is we get swabs, we take them in the lab, and then we take lots of colonies, even if they look identical, 6 to 12 colonies usually, and then we'll whole genome sequence all of them and look for exactly how many changes there are in their entire genome. And we've only looked, again, we have a focus on recurrent vaginal yeast infections. We do have some isolates from healthy people, but it's primarily this group. Way more variable than we expected. So I expected it to be very, very similar, but what we're seeing is actually a lot more variation. So what that means is that these populations are more stable than what we expect. Even if you take drugs and the population size goes down, they're not wiped out entirely. So they're probably a small population hiding out in your epithelial cells or somewhere where the drugs aren't reaching. And so they're maintaining this variability. And that's a problem because evolution requires variability to happen. And so if you have variation being maintained, you can then adapt faster to whatever happens next.
Hello, everyone, and thank you so much for this wonderful evening. I have a question specifically for you, Dr. Kumar. So you mentioned that the tap water quality is really bad. The question is how bad? In terms of the microbes or the non-microbial elements, and then on an individual level, how can we minimize the risks associated with the poor quality of water?
So the water quality I was talking about was that in indigenous communities. As it was mentioned earlier, actually Winnipeg tap water is one of the cleanest water you can drink off tap on this planet. So and yes, and the water actually comes from Shoal Lake 40, a community that did not have access to drinking water for 40 years, right? So we were getting water, but and the water is so clean that a lot of these bottling, the bottled water that we buy is actually just municipal water filtered. So just double check your bottle water next time you buy, and you'll see that some of that is actually coming from tap, filtered a few times. But in indigenous communities, this is a huge problem. So the work we did in collaboration with Dr. Fahrenhorst, we looked at the communities that actually have water treatment plants. So there are a couple of problems there. Number one is that a number of communities do not have water treatment plant. So they have no access to clean water. But then there are communities that actually have treatment plant, and it's working perfectly fine. So these are the communities that we worked with. The water from the treatment plant was absolutely clean, but as soon as it got into the distribution system, we started seeing E. coli, we started seeing coliforms, so these are fecal bacteria. And not only that, we also started seeing evidence of antibiotic-resistant bacteria. So what that tells us is that there is tremendous risk in people who are consuming that water. We didn't look at the health effects directly, but if you're drinking water contaminated with E. coli, I don't know, some of you might remember, I think it was about 7 years ago, in Winnipeg, we had a false positive test, and there was boiling water advisory throughout the city, right, for 2 days. And we were all panicking. There were people getting into fights at stores over bottled water for 2 days. Just imagine communities living like that for 40 years, right? So that's the problem that I was sort of alluding to.
Hello, everybody. So this is so nice. I work with all of you. I'm Kimia. I'm the comms lead at the faculty. And one of the problems I also come across is that we don't have enough people or enough hours in the day, for me at least, to write communications pieces for every research paper published or every research area. But also one of the other feedback I hear from the non-expert audience in science is that sometimes that audience doesn't feel invited in to science. They feel alienated by the way we communicate science or just by the idea of scientists. And I'm just wondering, what do you think, in your opinion, we can do to kind of improve that, to invite people in? I'm also totally going to use that in my own work. Thank you.
I'll take first stab at that question. I think so. And I'll criticize ourselves here. For the longest time, we've been doing science for scientists because that's what we were trained for. Things are changing. I think there is more communication from universities and research institutions for public. But, I mean, really, if you give me one of Dr. Pelka's papers to read, I probably would get 10% of that because it's written for virologists, and same thing the other way. Because we have always, I mean, one of the biggest criticism I have of ourselves is that we have, for the longest time, lived very happily in bubbles that we enjoy, right? And it's time, just based on what's happening around us, it's actually time to break those bubbles. And this is, and I don't think we scientists can do that by ourselves. We really need experts who can tell our story, because we are not good at telling stories to general public. So this is certainly something that we need. It needs to be a multi-pronged approach. I don't think you can, I mean, my colleagues might disagree, but I don't think you can rely on scientists to actually communicate with public. You actually need people who can communicate with public.
To add to that, you know, I do like my bubble. And it's, you know, it's very difficult to step out of that. It's, you know, writing something that is easy to understand for the layperson and explaining it is really difficult when that is not something you have ever been doing in your entire life, where you have to write scientifically dense publication like Dr. Kumar said. You know, I can read his paper, but yeah, I probably will get maybe 10 or 20% of it, and the other way around is true. So it's difficult. So presence of people like you, Kimia, you know, and others who can do that much more effectively is very important. I think the university has improved that. Certainly I've seen that since I started here. That has been growing and getting better. I've participated, you know, in quite a few. And you guys can make it happen. And I think it's important for the government, for the university, and for others to invest in that and make this sort of more accessible and easier to understand for the general public. And I think that's critical also in combating, you know, the misinformation, the TikTok influencers that don't know anything and just, you know, spread hearsay or whatever. And hopefully we can get better with time as well.
One other thing I would add is if you're someone who subscribes to the Winnipeg Free Press, which, by the way, is the jewel of Winnipeg that we do not appreciate enough, having an independent free newspaper in this city. If you subscribe to the Free Press, if you subscribe to the Globe and Mail, if you watch the CBC, advocate for science journalism positions, this is a whole discipline, and they have disappeared from mainstream media, and funding for science journalists is like at an all-time low. It's something you can do, all of you.
All right, this question comes from Raz, a alumni joining us in BC. Does any of your research leverage AI tools such as machine learning? And as a follow-up, do you find that you need to justify the use of these tools to funders when you are applying for grants?
Well, I'll take a stab at this one. Because that was actually a big part of our big CBRIF application for the viral vaccine vectors that we got funded to the initiative. So we do use it, but there's actually a very good reason why we decided to use it. We're leaning more towards machine learning rather than AI itself. But what we are trying to do in a nutshell is take a bunch of different vaccine candidates, put them all through all sorts of immunological tests, test those vaccine candidates in animals to see which ones perform the best, and then combine all of that data to see which early immunological tests can provide good predictive value of an effective vaccine. And I think in uses like this, machine learning can really save a lot of time, where it can take a lot of variables and condense them to the critical ones that are good predictors for a potential good therapeutic or a good vaccine. And that can save time and money, because these types of investigations, vaccine development, is extremely time consuming. It's also very expensive, and nobody wants to invest in it because there's not a lot of money to be made at the end. So if we can reduce the number of tests and the time it takes, we can successfully develop an effective and safe vaccine, with our goal being about 100 days, and that way we can leverage those machine learning and potentially some AI tools as well that can expedite that research and pick out those things. Because AI and machine learning is really good at identifying these patterns and other things that are much more difficult for humans to do.
Maybe I can add to Peter. This is just tool, and I think it's legitimate to use this tool. In my research, when we are making synthetic enzyme, this is combinatorial problem. And now it's the question whether you ask your student to screen billions of bacterial colonies and then spend six months only to pipetting every day, 8 hours, or if you have language model that can simply solve this combinatorial problem, and your student maybe will work only 2 days in the lab. It's just useful tool, and we should use it. Of course, like everywhere in science, using tools like AI, using genetic engineering, so also need good governance. And I think we are quite good when it's about governance. The general problem, not only AI, is that, for example, in my research, when I'm making patents, you often have scientific progress to such a level that legislator following us, sometimes decades, sometimes 2 decades, is behind us. And this is again problem of communication between different branches. Yeah.
Hi again. Okay, so this one is for the evolutionary biologist of the group. What's your name?
Aleeza.
Hi, Aleeza. I was asking for your last name.
Oh, Gerstein.
Okay, Dr. Gerstein. So I remember it was mentioned that bacteria, as of recently, as part of the rise of antibiotic resistance, have gained the ability to release, I guess, interceptive proteins in order to, it sounds like, neutralize any antibiotic particles in, like, serum, right? Or I guess in the environment. Is this ability, like, a new development in the microbial world, or what was the original evolutionary purpose of this mechanism?
So maybe I'll take that one. So antibiotic resistance is not a new mechanism at all. So there is microbial competition in nature all the time. A lot of fungi produce different antimicrobials, and the idea is to gain advantage over their neighbors. So there have been studies, and actually these are Canadian studies. So, shout out to some of the Canadian researchers out of McMaster University, where they looked at permafrost. They looked at some caves in New Mexico that was sealed for millions of years, and they found bacteria from there, and they carried antibiotic resistance genes. So antibiotic resistance is not new. The pressure that antibiotics are putting in the environment is new. The scale of that pressure is new, right? So if a fungi is producing antimicrobial, it's in its vicinity, it's only damaging cells or inhibiting cells that are close to each other. What we as humans are doing is literally taking antibiotics in a spray tank and going around orange orchards and spraying it on trees, right? So it's just the scale of this. And You know, we were talking about if something is too clean. Next time you're grocery shopping, just for interest, look around and see how many things are being marketed as antibacterial. And why? Why are we paying $2 more for a toothpaste that is antibacterial versus the one which is not? Why do we need that? And we don't need that. But what this is doing is you're using that toothpaste with antibacterial, you're spitting in your sink, that's going in your water, that's going in the river, that's going in the lake. So all these compounds are now, so there was a study actually carried out, one of the common compounds in these products is called triclosan, and there was a study carried out in the US, and they tested urine of adult males, and 30% of adult males in that study actually had triclosan in their urine. Where is that coming from? We don't produce triclosan, right? So the scale is the problem. What we are doing as humans, and this is, I think, one of the problems. When we get good thing, we don't know how much to, you know, where to contain ourselves. And so the problem is just the success of antibiotics has been a problem.
Maybe just a quick, since we're doing science communication here, one of the things that drives me nuts is when I'm in a bathroom and I see antimicrobial hand sanitizer beside the soap. Because something that works incredibly well is soap. Pardon me?
If you use it.
Soap is excellent. And if you have a choice between soap and antimicrobial hand sanitizer, use the soap.
So we've talked a fair bit tonight about science and communication. And I'm just not wondering if we need an advocate for that. Dr. Duckworth was an excellent advocate for physics and mineralogy. David Suzuki, of course, is an advocate for ecology and so on. And I just wonder if, as a group of scientists, whether your organization or whatever has sought out an advocate. My own experience as a student, as a researcher, and so on, was that my advisor said, I want this information, this work to be spread around. Would you mind if I have someone look at it? Turned out that the person reviewing that work was the president of the NDP party. She said to me, do you mind if I show this to the premier? And in a week and a half, the premier had shut down the initiative that I had raised concern about. And, so I had an advocate and went in the right direction. And, communication is so important. In the political world, if there's a problem, get the communications people involved to sort it out and so on. Is that something that you could be encouraged to do?
I think we need science advocates who are elected officials. I think that really requires that. I know in recent years, there's been a big push in the US to get scientists to run for office. I haven't really seen a similar thing here. Although we have occasionally had scientists in office, it's very, very rare for a myriad of reasons. I think that's the policies need to change, and so you need someone to listen.
To be fair, the university has improved, like I said. Certainly I've seen it over the last 10 years or so, 15 years that I've been here. I think the problem is scientists themselves are too busy to get into politics and often don't want to be bothered. And on the other hand, the politicians are too busy with the government, running the government and other things that they can be bothered, unless it's a big, real, serious problem. Yeah, measles or the pandemic or whatever, right? But so far it's baby steps. The university is better. I've done a lot of outreach as well, videos. My YouTube video on vaccines had pretty good outreach. So, we're working on it and trying to improve as well, but it is difficult for reasons you mentioned, we mentioned, and many others as well.
So we're at the time that we will take the last question. Is that or are we done? Okay, I think we have reached the time limit. So thank you all for joining us. It was lovely to have you all here, and I hope that there was some level of value that you gleaned from the discussion. So thank you once again.